Alright guys, welcome back to Underground Science. In this video we're going to be talking about processing of pre-mRNA, okay, and also splicing of pre-mRNA, or the splicing of the introns and exons, okay. So let's start with the processing of pre-mRNA. So what we just made is pre-mRNA, right? And so we just left off making this pre-mRNA strand in the last video, right? And we said that we have a 5' untranslated region, a 3' untranslated region. Okay, in the 3' untranslated region, we said we have that polyadenylation signal. Okay, so first thing what happens in processing of pre-mRNA is we have to add a 5' cap. Okay, so what does that look like? So a 5' cap is also called a 7-methylguanosine cap. Okay, and there's three steps to this. Okay, so first we add a 5' cap by taking RNA triphosphatase. This is an enzyme, okay? It's going to break the bond between the second and third phosphate. Okay, so it's going to break this bond. So then we're going to end up with only 2 phosphates here, right? So that's RNA triphosphatase. Then we're going to have guanylyl transferase, okay? Guanylyl transferase is going to put a 7-methylguanosine onto this phosphate. Specifically, it's going to be, I skipped ahead, right? A guanylyl transferase, all it's going to do is it's going to put a GMP group on this second phosphate. So what we can do is write that down, right? It's going to put a GMP group, okay, on that second phosphate, a guanosine monophosphate group. Because what actually happens is guanylyl transferase will actually try to put a GTP on it, but what happens is GTP gets broken down into GMP. And then, so what is this? This is guanosine triphosphate. This is guanosine monophosphate, right? So the difference is 2 phosphates. So that means we lose a pyrophosphate. So then GMP will go ahead and bind onto the second phosphate. Okay, so then we have a GMP now. Then the last step is a methyl transferase will come in and put a methyl group on this GMP. So we're going to have a methyl group right here, okay? So this whole thing now, what we just did is we just added a 5' cap. Okay, this is the 5' end, and we just added a cap. Okay, that's why it's called the 5' cap. Next, what we're gonna do is we're gonna add a 3' poly-A tail. Okay, so it's exactly what it says, right? We're gonna add a poly-A tail, meaning we're gonna add a segment with around 200 adenines, okay, on the 3' end. Okay, so it's basic. That's why we have this polyadenylation signal, because this helps poly-A polymerase to come in and basically add a bunch of adenines. Okay, so it's going to be around like 200 adenines. Okay, so we can even add more, right? That's basically why the polyadenylation signal is important, because it's going to help poly-A polymerase come to the 3' end. And near this polyadenylation signal, poly-A polymerase will add around 200 adenines, okay? Now, why do we do these steps, the 5' cap and the 3' poly-A tail? Because it basically it prevents degradation of this pre-mRNA that we just synthesized, right? But after we just added the 5' cap, right, and the 3' poly-A tail, now this is one step closer to becoming mRNA. Okay, it's not mRNA yet, it's pre-mRNA right now. Okay, so remember, this poly-A tail and the 5' cap is good for preventing degradation of this pre-mRNA. Okay, and it helps export this export this RNA strand from the nucleus into the cytoplasm so it can undergo translation. Okay, so that's processing. Okay, now we have to do splicing, because remember what we said is in the open reading frame, which is after the 5' untranslated region and before the 3' untranslated region, in the open reading frame, we have exons and introns. Introns cannot get translated to proteins. We have to take these out. Exons are the ones that stay, okay? So how do we splice the introns out, right? So that's what we're going to talk about next. In fact, what I was thinking is let's talk about splicing in the video right after this, okay? So this video is mostly about RNA processing. Now let's talk about splicing in the next video. Okay, see you there.